Hi everyone, in today's video we're going to talk about proteins, and we're going to start off discussing what are called the levels of protein structure. And these are classified in the following way. 1 superscript 0, that's primary structure. Okay, as you might imagine, 2 superscript 0 is secondary. Tertiary, and then quaternary. So what these basically describe is sort of a progressive amount of structure that can be thought of when you start talking about polypeptides and how they end up folding when you put them into an aqueous environment. So first off, primary structure is just the linear sequence of amino acids. Okay, so if you envision taking a long polypeptide chain, let's say it's a protein with 300 amino acids, if you were to grab the two ends, the N and the C terminus, and literally stretch that out and sort of linearize that long peptide, you'd then have a sequence of amino acids going from the N terminus to C terminus, described as the primary structure. Secondary structure are then locally folded patterns. And the best examples are the so-called alpha helix or the beta sheet. We'll talk about what those are in a bit. But these are small local areas of that long polypeptide chain that when you put them into water will sort of inherently like to fold into these certain small local structures. Tertiary protein structure would be the fully folded and active protein. I like to include this and active part because in order for a protein to actually do its function, it has to fold up in the correct way. And there certainly are some times when a protein misfolds and is not actually active. So if a protein has full tertiary structure, it's going to have a lot of these secondary folded patterns, alpha helices, beta sheets, and other local patterns. But essentially now that long linear polypeptide will start to fold up and compress into the final shape of the fully folded protein. And then finally, you can, under certain circumstances, have what's called quaternary structure, which is actually the interaction of 2 or more polypeptide chains. So, for example, the protein hemoglobin, which is in our red blood cells, hemoglobin is composed of actually 4 different proteins that form a tetramer. And each of those is fully folded. We have 2 alphas and 2 beta polypeptide chains, but those each individually fold up and then they aggregate to form this larger, what's called tetramer. And that is an example of quaternary structure. Now, not all, just so happens that this one is a tetramer. Just because it's called quaternary doesn't mean it has to be a tetramer. These can often be hexamers or dimers. They just try and envision, a polypeptide chain that's folded. Now, 2 of those or more coming together to give a more complicated folded structure. So, what is it in proteins that actually causes these to fold? Well, let me list out some of the major attractive forces between amino acids that you would find in proteins. And I'm going to list this in order of sort of decreasing strength. Okay, so the first one that I'm going to mention is what's called a disulfide bond. And as we saw before, these arise when you have 2 cysteine residues that, if you envision them sort of far apart on the linear sequence of the peptide chain, if they were to come close to each other, they could form an actual covalent bond. So this one is actually the strongest of them because it is covalent. And in essence, you can think of that essentially making now a long linear chain, forming now a loop. And if a protein has multiple cysteines, you could form potentially multiple disulfide bonds. So that's going to be the strongest of these types of forces. Next are the so-called ionic bonds, okay, or ionic interactions. So for example, if I had, let's say, the amino acid aspartic acid, and I'm drawing it as its physiological protonation state, which would be a carboxylate. Let's say that was somewhere on one end of a protein. And maybe on another end, let's say I had one of the basic amino acids, let's say lysine. Okay, so now if I bring this positively charged lysine in close proximity to the negatively charged carboxylate, this now can form an ionic bond. And that's a fairly strong interaction. Now those are often called salt bridges in a protein. Next would come hydrogen bonds. So if you had something, let's say like a serine in one part of the protein, and then somewhere else in the protein, let's say we had, let's draw, okay, this is asparagine. I'm sorry, yeah, asparagine. This could now form a hydrogen bond in this way between the carbonyl and the OH. Alternatively, you could potentially have a hydrogen bond between the oxygen of a serine and the NH of asparagine. So hydrogen bonds individually are fairly weak. However, a fully folded protein that exhibits both secondary and tertiary structure can have several hundred, maybe even 1,000 different networked hydrogen bonds that help sort of hold the fully folded structure together. Lastly, we have dispersion forces. London, call those London forces. Or in the context of proteins, these are often referred to as what's called the hydrophobic effect. Okay, because proteins, when they fold, they're surrounded by the solvent water, the exterior of a protein tends to have amino acids that can easily hydrogen bond with water, whereas the nonpolar amino acids, things like leucine or phenylalanine, they tend to become embedded in the core of the protein structure. And because of that, because of the close proximity of those nonpolar groups, they do exhibit dispersion, London dispersion forces, but this is often referred to as the hydrophobic effect. So if I had, let's say, a leucine molecule, and then maybe a phenylalanine, okay, if those two on distant parts of the amino acid chain, if those ended up close to each other in the fully folded structure, there would be a slight attractive force between them. And again, in proteins, that's referred to as the so-called hydrophobic effect. So I briefly want to talk about the two most common secondary structural elements that you'd find in proteins, those the alpha helix and the beta sheet. I'll start with the alpha helix. What this is, if you envision taking that long polypeptide chain and allowing a small region of it, maybe between 10 to 20 amino acids, to sort of form a corkscrew-like structure. It's going to look kind of like this. And I'm just drawing, say, the peptide backbone. I'm not showing all of the side chains here. Let's say this was the N terminus, this is the C terminus. Certain sequences and certain amino acids like to form this type of corkscrew-like helical structure. It also would sort of, if you envision sort of a spiral staircase, it has kind of that look to it. And what happens here is you have NH groups that are then hydrogen bonded to the amide carbonyl group about 3.5 amino acids away, okay? But you end up forming these throughout the entire structure, okay? So you end up getting multiple hydrogen bonds forming along the spiral staircase, if you will, of the helix, and about 3.5 amino acids per turn of the helix is what it works out to be. But these alpha helices are very, very common in protein structures. And again, they form between these intermolecular, within the same molecule, hydrogen bonds. One other thing that I'll note here, the side chains, so the R groups coming off each different amino acid, those radiate out away from the central axis of the helix. So if I were to now sight down from the top of this helix, if I were to redraw this, the R groups actually radiate out around the helix. And there would then be a very, very small opening or pore going sort of straight down through the middle of that helix. Now it is quite small, but that's the overall structure of an alpha helix. They can be between 8 amino acids up to maybe 40 or 50 amino acids. They can be quite long. Beta sheets are quite a bit different. These tend to exist if you imagine taking that long polypeptide chain and allowing certain regions of it to simply fold over back and forth, kind of like an accordion. You can then have hydrogen bonding interactions between carbonyls of an amide on one part of the protein to an NH group and vice versa. Okay, where we have little hydrogen bonds here. So what ends up happening is you have multiple hydrogen bonds sort of holding this folded structure together. And if I were to look at this from the side, beta sheets often have an accordion look to them. Okay, where I have hydrogen bonding interactions holding these together. And it turns out that the side chains in beta sheets actually tend to alternate faces or sides of the sheet. Okay, so if you envision now looking at this from the side, one face or plane of a beta sheet, every other side chain is sticking up, and then every adjacent one is then sticking down. So you can actually end up with a situation where you have beta sheets where one surface might be coated with, say, polar amino acids, things that can hydrogen bond easily, whereas the other surface is very hydrophobic, and you end up getting two different surfaces on a beta sheet. So in secondary structure, you would then have these small local folded patterns. So to wrap up our discussion of proteins, let's just briefly talk about tertiary structure. Now, tertiary structure, again, is the fully folded and active protein. So if I had a combination of alpha helices, I'm just going to draw this in a very crude schematic way. Here's an alpha helix. And let's say this is my N-terminus of my peptide chain. And then maybe there's a region that actually forms a beta sheet, something that looks like that. Then I might have another alpha helix. Maybe there's another turn, maybe another alpha helix, and then maybe another beta sheet. Now this is very crude looking, but and all I'm showing here is the backbone, and this would be then the C-terminus. But this might be a protein that, say, has 300 amino acids. Again, when you put it into water, these will spontaneously fold to form these local regions, and then ultimately those tend to collapse and form a fully folded structure. And in order for a protein to have its activity, okay, it must fold correctly. Okay, and sometimes even a single change in just say one amino acid, if it's at a critical location, if that particular amino acid happened to be changed, it could dramatically alter the folding of this protein and therefore affect its function. Numerous examples of this. The proteins involved in Alzheimer's disease, those involve a misfolded protein. Sickle cell anemia is another example that's brought about by misfolded globin proteins in hemoglobin, and those arise because of a single mutation at a single amino acid. All right, so that covers the sort of breadth of the primary, secondary, tertiary structure of proteins. A little bit later on, when we talk about oxygen transport, we'll get a little bit more into quaternary structure. All right, I hope that helps.